I'll do this in English. My name is Torsten Fraltoft, the CEO of AcouSort. AcouSort is a spin-out company from the University of Lund, from the group of Professor Thomas Lorel, who's the world-leading expert on what's called acoustofluidics. And what is acoustofluidics? Much more easily explained, it's sorting cells with sound. Before I do my actual presentation, I'd like to show you a small presentation video that we did half a year ago when we did our IPO at AcouSort. We were listed 9 January. I'll come back to that. But let me show you this presentation video. And as it was made for the emission, don't worry about at the end you are invited to participate in the emission. Interpret this as an invitation to take a closer look at the share and maybe make it part of your investment portfolio. So please enjoy this two-minute video. Let's see.
How can exposing cells to sound save lives? People suffering from lethal diseases have to rely on fast and accurate diagnostics to find and monitor the disease. The more we know about their specific condition, the better our chances are of curing it. However, diagnosing diseases is often a long and tedious process. Why? Because first, you need to find, identify, and process different cells or molecules from the body that contain the relevant information. Everything we need is typically present in a simple blood sample, but the information is rare and hard to find. Today, there is a big need for fast and automated methods. If only processing could be done more efficiently, diagnostics would be faster and many lives could be saved. So, it all comes down to sorting out the relevant cells and molecules, and that's where AcouSort comes in.
We have developed a method for instantly sorting out biological cells of interest by exposing them to sound waves.
Different types of cells have different size or density, and therefore the cells react differently to ultrasound. And this way we can separate the cells of interest from other cells, either by separating them in channels or by trapping them.
This will give us the required amount of substance that we're interested in, just enough for a diagnosis.
The method can be practiced in AcouSort's own developed research instruments, AcouTrap and AcouWash, and now AcouSort is also developing a plug-in module for building into existing commercial diagnostic systems. This way, the technique could be implemented by almost any diagnostic system provider without having to rebuild the entire system. Fast and accurate diagnoses save lives, and now you have the opportunity to be a part of this revolution. During fall 2016, AcouSort carries out a new issue of shares ahead of a planned listing on AcouSort. AcouSort, sorting cells with sound.
So, a little introduction, but let's take a closer look at some of the biggest challenges that we have in the healthcare sector of today.
How can
Now we are going too slowly. One of the top killers that are out there is sepsis or blood poisoning. Alone in the US, 250,000 people die every year due to sepsis. And what's even worse, this number increases 8% per year, every year. It's an enormous problem, and it's growing. So it's almost as big as cancer. As you can see in the lower right corner, lung cancer, breast cancer, and prostate cancer is more or less the same as the casualties you have in sepsis. So cancer is another very big challenge. This is even bigger than sepsis, as almost 500,000 people die every year alone in the US, 12,000 in Sweden. This also grows every year. not quite as fast, 2.5% per annum. So the very good question is, can cancer or sepsis actually be cured? And as most of you know, yes, they can, but only if they are diagnosed or found early enough. That's the challenge. Let's take a look at how you actually diagnose sepsis or cancer today. Well, sepsis is a matter of bacteria in the blood, so you have to take a blood sample. However, you cannot actually identify the bacteria right away. You have to culture them, multiply the bacteria, so you get enough for actually to actually characterize them. Then you can do mass spec or DNA analysis to identify the type, and then give the patient the right optimal treatment. For cancer, there's no treatment before you actually know where is the cancer, what is it, and how do you find out? You have to identify a tumor. This you do by CT scanning usually. Then you take a biopsy, a physical sample of the tumor, if you find it, and then you can do manual or semi-automated microscopy to actually find out which type it is, how it works. But in both these two cases, in a simple blood sample, you have the biomarkers that's needed to actually identify the condition. But the problem is that today there are no effective separation methods that can actually give you this information right away. This is where acoustic separation or acoustic trapping comes into play. So let me talk a little bit, because seeing is believing, and show you some small videos of how this separation actually works in the lab. Here I'm showing a chip where we have made a small flow channel. It can be done in silicon or in glass. We use glass in AcouSort. In the lower left corner, you see such a cross-section of a channel in a glass chip, and all the black dots there are cells or particles. When you turn on the ultrasound, then you can focus these particles to a pressure node in the middle of the channel. So that's the sort of most fundamental element. So why do you want to do this in connection with cell handling? It's a very generic technology, it's versatile, and what's even better, it's harmless to the cells. So the cells, they move around or are manipulated, but it doesn't do any harm to the cells. It's quite rapid, and you can do quite high throughputs, because you can do this in a continuous flow. As the cell passes by, you can manipulate them. Here, on the next small video, you see how you can concentrate cells. This is a flow channel with a Y, and if there's no sound, the particles will flow all over the place, but if you turn on the sound, you focus them, and you can get them concentrated in a smaller part of the liquid. You can also do separation. In this video, you see 5 micron red ones, 7 and 10 micron particles, and by turning on the right the sound and adjusting to the right level, you can actually separate them in different exit channels. And also, you can do cell washing. Cell washing means that you have cells in one buffer, and you want to change them into another clean buffer or different buffer. This is done here at the bottom. Inlet, the cells are actually in the dark blue liquid at the edges, and then when you turn on the sound, you focus them into the middle where you have a clean, clear buffer, as you can see. 120 million Swedish crowns have been invested in this research since 2009 in Thomas Laurel's group or collaborating groups, and as of today, 30 researchers are currently working under the umbrella of Thomas Laurel in various projects. We talked about cancer and the circulating cancer cells that are actually present in the blood when you have one of the most common cancers. If you can get those cancer cells out of the blood, you can have kind of a liquid biopsy. It's like a test sample of your cancer without actually going in and finding the tumor. This can be used to guide your treatment, to find out how to treat the patient. But it can also be used to monitor rapidly how a given treatment actually works. I mean, how the response by the body is. And of course, it can be used as a support of new drug development. In this video here, you see 5 and 7 micron particles being separated. And that is exactly the difference between white blood cells and tumor cells in the blood. They have roughly this difference. So as you can see, it's possible to actually separate those two. In the real world, there's only one cancer cell among 1 million white blood cells. So it's difficult to find them, and this is still only at the research level, not a final product. My last technical slide has to do with centrifugation. What happens if, in a view, I'm sure everyone has tried to deliver a blood sample at the clinic. What is the first thing happening when doing that? That is, at the clinic or at the laboratory, somebody will centrifuge your blood sample. Why is that? Because, as you can see on the panel, you would like to get access to the blood plasma. The plasma is that clean liquid that the white and the red blood cells are immersed in the blood. I mean, the blood appears red because of the red blood cells, but between the liquid where these cells are floating is a clean plasma, as it's called. And 99% of all blood analysis, they are actually taking place on the blood plasma. So you want, or you need to get rid of all the blood cells to do this analysis. This is, of course, very easy and fine if you have a laboratory and a centrifuge right there. But if you wanted to do the same type of analysis in the field, point of care, at home, away from the laboratory, then you need a technology to do this separation. And this is also where acoustophoresis comes in. As you can see on this video, this is whole blood entered into such a small chip. And if you turn on the sound, ultrasound, then the blood cells are focused into the middle and the plasma will come out at the sides. Exactly this this last application is something where we work with 3 major life science companies to develop OEM products, meaning small module that can do this separation in their analytical systems. I'll get back to that. We've, of course, talked or looked into the market for this. And we've tried to put different applications into whether it's very large, whether it's large or medium or lower. Now, some of the things I've talked about just before, the circulating tumor cells is a very large market. Bacterial concentration to diagnose sepsis, also quite large market. Plasma isolation, as I just mentioned, also very large. Particle washing and extracellular vesicle concentration, that is also elements we are working on in the company. But besides these initial applications, there are numerous more applications. So we are not going to run out of business opportunities in the near future, at least. So let's take a look at the initial products that we have in different stages of development. First, there is the AcouTrap system. It's a particle trapping system which can enable bacteria and microvesicle research. This instrument has already been launched, so the commercialization is already in progress. We are currently showing the system at various big scientific conferences to make the relevant researchers aware of this new technology. And we have already sold 4 systems. The AcouWash is a cell washing station. So here you can actually take a sample with cells and exchange the buffer to a new clean one, or you can concentrate the cells to a higher level. This is still in its final product maturation, and we hope to be able to launch this for sale late this year. Then at the end, we have the blood plasma separation module that we developed together with so far 3 large life science operators. All these companies have analytical systems already in the market. They are really, really big companies. I've previously said they are 50,000 employees. I have to say it's only 2 of them there are. The other one is a little bit smaller. But they are very big, and they have large market shares of different application areas. They all would like to include the separation technology in their existing or soon to become released products. And we work with 3 of these companies to do this. So our commercialization strategy is twofold. The main strategy is to develop and commercialize such OEM products, so modules to build into other people's systems. And this we do in alliance with the global life science system providers. This can provide direction, so we are sure to develop something that's really useful, and at the same time, the collaboration will provide some financing of this development. Meaning these strategic collaboration partners actually pay AcouSort for at least part of the development. At the same time, we also sell generic tools, the benchtop models that you just saw, the AcouTrap and the AcouWash, to researchers, so they can practice using acoustophoretic separation or trapping in the lab. This would generate early revenue for the company, and also it can contribute to developing new strategic partnerships if these researchers are from private industry. The commercialization status for the three product groups is then blood plasma OEM module. We are working with the 3 global partners as mentioned, and we hope to be able to launch the first product late 2018. Of course, this is not only up to AcouSort, because we are working with partners on this, but you could say the module as such can be launched from our point of view late 2018, the first version. And then, of course, it needs to be built in, and it needs to be approved for different applications by the partner afterwards. For the research instruments, we, as I mentioned, already launched the AcouTrap, and are just now in the process of building 10 systems. These we anticipate selling during the rest of the year, and then we are planning to sell 20 of these systems next year. AcouWash, still in final development, so we are planning to launch this towards the end of this year, hopefully selling 6 systems at the same time, and then planning to sell 10 systems next year. The people behind the company, here we have the board of directors. Martin Linde is the chairman. He's a very experienced business executive in Sweden, CEO and president of what's called the Nordic Food Group. We have Thomas Lorell, who is the key founder and is the professor at Lund University and representing the core technology. We have Stefan Scheding, who is a medical doctor, and he's our link to the clinical world. So he's a professor at Lund University, associated professor in hematology. And then we have Annelie Trøtson, who's an experienced lawyer at the board. But the operational team, coming up, I hope. Try again. Here. We have the original team that, besides myself, includes Pelle Olsson, Mikael Evander, and Per Augustsson, who are all coming out of the university group at Lund University. And then we have the new team members, because after the IPO, we have been able to increase the team to make it, you could say, a real commercial organization. It includes Lars Leukner, who's our new Director of Sales and Marketing, a very good expert in customer relations building. And then Magnus Hivert, who is a productization manager with us. We need expertise in transferring these applications and these scientific results into real products, and he is the person to do that. At the end, I would like to just go through the progress that we've made since the IPO in January. It's exactly today 4 months since we had the first listing. And I have to say we were very pleased with the share emission that we did in December. It was 697% subscribed. And also the IPO, the listing in January, went really well. We can take a look at how the share has been doing. We had the emission at Swedish crown 550, and already the first day it went up by 60 or 70% to 850, and has been fluctuating back and forth around 10 crowns since then. And volume has been 8.8 million, so 8.8 million shares have changed hands, and that is pretty good since the company only has 7.5 million, and more than half of these are locked up so far the first year. So there has been a lot of transaction, more than 10,000 deals, actual deals at AcouSort. And as mentioned, the share price is up around 80% compared to the emission price. But besides that, we've put the new organization in place. We've hired 2 extra in the management team and a few extra people in the other operational part of the company. We have increased the market strength presence in the company by going to 3 very big conferences and exhibiting the AcouTrap system. We've actually also managed to get our first international patent issued in EU and the US. And then we have increased the number of strategic partnerships from 2 that we had in the fall until 3 now. So we have 3 ongoing strategic partnerships for OEM products. And in general, all these 3 OEM collaborations developed really well. So that was all I had to present, and thank you very much for listening.
Tack. Har vi någon som vill passa på och ställa en fråga till Torsten medan han finns tillgänglig här? Nej. Torsten, then I would take the opportunity.
Sure.
Is there any particular geographical area where you foresee those sales to come within this year and next year, or is it, for example, within Europe, or is it all over the world, or do you focus upon the Scandinavian market, for example?
We are focusing on the North American and the European market. We will so those sales we are talking about will appear in one of those 2 regions. You can say we are targeting a quite narrow research group for this first product. So we know more or less exactly who they are, and during this last period, we've experienced that all the big pharma companies in both US and Europe, I mean the top 10, they have ongoing research projects, in this field of extracellular vesicle research. And that's exactly where the AcouTrap will go in and make a difference to separate out those extracellular vesicles from body fluid samples of different kinds. And that's a very hot topic. But of course, there are maybe in the world 500, 700, 800 groups working in this field, and those groups are the target for the first product.
Okay. Okay. Any other question? No. Where do you meet your potential customers? I guess since do you know who they are? I guess you are the ones to visit them, and you approach them, and you explain the benefit that they will get by using your product. Is that correct? Understood?
That's going to be the second tier, because since this technology is very new, the first customers are going to be the so-called early adopters, and they will actually come to us.
Okay.
How do they learn about it? That's at these 3 big conferences that we're going to. Specifically, one of them is dedicated entirely to this specific group of customers. So you can say 100% of the people coming to this are potential customers.
Okay.
And the people we meet there, some of them will be the early adopters and will buy the first 10 systems. We're not in doubt on that. Then, as the rumor spreads that this is really successful and people has a lot of benefit from using the product, then we can go out and knock on doors on the next ones that we know from the public literature are working in the same field.
Okay, I see. Okay, anything else you would like to add before we finish here?
I just wanted to thank everyone who has been supporting the company for the last 4 months, and thank you for the trade and for the long-term participation. Thank you very much.
Tack. Tack så mycket.